Okay, hi, my name is Dr. Anne Cunningham and I'm an immunologist. I work in the University Brunei Darussalam, which is on the beautiful island of Borneo. This time last year, I became transfixed with the numbers of cases of this new virus coming out of China. I think everybody in Southeast Asia was very alarmed because of their experiences with SARS. So alarm bells are ringing. And so a very simple idea, if there's no treatment for a new virus, is to use something tried and tested. And that idea was to passively transfer preformed antibodies from patients who had recovered. And that is the convalescent plasma. So in the last year, a lot of work has been done. And some has shown that there is benefit, particularly if it's given early on. You can follow this in other places. So there is a living systematic review with the Cochrane Library. And on VUMEDI itself, Professor Joyner gave a very nice presentation just on the end of January. And he's also published some of the early data from the study in the US. So a lot more data will come out, and probably it helps some people, particularly if it's given early on. So what's the next obvious idea? So obviously, when you make a natural immune response, we have a very diverse immune system. So a virus will stimulate many different specific antibodies. And what we make is a polyclonal response. And that's good because it helps us to get better. But maybe it's more interesting to actually take a single preparation of highly purified antibodies and to use those instead of using convalescent plasma. So what's the difference and why might that be an advantage? If we think about clearly you can clinically obtain convalescent plasma from patients, but you need to make sure there's no other infectious agents. You want to make sure that there's no procoagulation or pro-inflammatory factors in there. And of course, each batch you get is going to be different. So some might be high titer, some might be low titer, you don't know. So it's very inconsistent and difficult. So therefore, it's going to be attractive if we can make monoclonal antibodies where we have a renewable source of high purity and we can really be clear what exactly is the specificity. Because when we come to the good of this presentation, antibodies are fantastic. So they are amazing effector molecules. They can recognize and destroy pathogens. So they have the two domains. This is the basic structure of an antibody. You can see there's two identical heavy chains and two identical light chains. Within every antibody, there is a fraction, the antigen binding domain. So antibodies have two identical antigen binding domains, and then they exert their biological function through their Fc domain. And those Fc receptors, those Fc domains bind to Fc receptors on a whole range of other effector cells. When we talk about COVID, we mainly talk about the antibodies blocking entry. So we have a lot of experience of antibodies. Monoclonal antibody technology was invented in the late 1970s. We can create antibodies and characterize them, and they've really transformed research. They're widely used in diagnostics, and really for the last 30 years, they've also been increasingly and widely used in therapeutics. So the so-called advent of biologicals. In the 1980s, a new discipline called antibody engineering came about. And so obviously that's quite a mature discipline now. So the first antibodies were made in mice. So then they were humanized. You can take just the single variable fragment, single chain variable fragment. So this part here. And also, interestingly, not all mammals make antibodies look like this. So camels and alpaca make something called a nanobody. And a nanobody is essentially a smaller version of an antibody. And there may be some advantages in having a small size, so they don't get blocked in when they make an immune complex. Okay, so antibodies are made by B lymphocytes. In fact, they are the released receptor. Okay, and one of the great advantages is that our adaptive system is highly diverse. We can recognize anything specifically. Once we've seen it before, we have memory and we make an escalating response. So the part of the antibody that actually binds are these what we call hypervariable domains right at the very end of the molecule. And in particular, we have these complementary determining regions that create the three-dimensional shape or pocket that gives the specificity to a particular antibody. So it's like a jigsaw puzzle or a lock and a key. And that shape will determine exactly what part of the pathogen, which we call the epitope, gets bound to by a particular antibody. So in terms of antibody engineering, when you look at antibodies that are approved for human use, you'll see different ones. So you won't see so many human ones, murine ones anymore, because obviously we could make an immune response to the murine antibody and that would be harmful. So these can be humanized, where we put the structural part of the human antibody in, or we can even engineer just the very small sequences that make up the pocket. So they're going to be much less immunogenic. And now there are human immunoglobulin genes in mice, or in the case of recovering from an infectious disease like COVID, you can take human B cells and make human antibodies. And we have the technology to do that. So I'm not an antibody engineer, I'm just an immunologist. But just to give you a kind of very simple picture of the various different kinds of antibodies that are used. So the first antibodies really that were approved for human use were in transplantation in the late 1980s. And this is just a kind of recap showing you that most antibodies are used for oncology and autoimmunity. But you can see that there's always been a number of antibodies that have been used, monoclonal antibodies, used for infectious disease. So this is a bit like the passive transfer of preformed antibodies, except that in this case, we have very highly specific, purified, renewable source kind of antibodies. So these are the current antibodies that are already approved for use against different pathogens. So you can see that they are IgG1 and they are humanized or human. And this is interesting here in 2020 against Ebola. There's a mixture of 3 human IgG1, and I'll come back to the benefit of adding mixtures later. So I just want to show you an image. So let me switch screens and just play this briefly. So this is showing you the spike protein of SARS, and this is a monoclonal antibody. So you can see its antigen binding domains. So you can see the spike protein going into the trimer has a different up and down, and the antibodies are binding to the receptor binding domain of the spike protein to block its entry into the receptor on our body cells, the ACE receptor. So this is what is happening when you have a neutralizing antibody that will block a virus from binding to its receptor. So obviously, that's very attractive. So where are we? Where are we up to with this next logical step? So there are 2 preparations that have been given emergency use authorization for use. So one of them is the Regeneron cocktail, which was used by the former President Trump, and there's also another one from Eli Lilly. When you look, if you're interested in antibodies, a very good resource would be the Antibody Society, and they have a spreadsheet of all the biologicals that are in the pipeline. And in that list, there are 123 different monoclonal antibody preparations to the SARS-CoV-2 virus. So the most advanced ones are, so there's some data for this neutralizing antibody cocktail. So just recently in the NEJM, so there was a randomized multi-center RCT. So what's interesting about this, so the numbers are quite small. They had 228 patients, and of those, about 45% were antibody positive and about 41% were antibody negative at the start before randomization. And their actual primary outcome they were looking at was medically attended visit. But they also looked at viral load and a whole range of other things. What they found was that patients who had the highest viral load had the largest benefit in treatment. And if they broke their analysis down into the patients that were either antibody positive or antibody negative in the different groups, then you could see that if you are antibody negative, you had higher virus and you benefited more compared to the placebo from the treatment. And that treatment was seen by day 3. So if they were given the infusion on day 1, by day 3, they saw the biggest decrease in viral load. So that kind of makes sense. But what they said was most infected people recover because of their immune response. So other antibodies that are in phase 3 studies and have been well described are this list here. So what about the bad? So antibodies, very powerful effector molecules, but viruses have been avoiding recognition and avoiding destruction in order to infect us forever. So we've noticed that, and there's been a couple of preprints. So this means that they have been put on a preprint server. They haven't been peer reviewed, but I've just extracted some of the data to show you. So there's a study from South Africa, and you can see there is a difference in the assays when they look at the response of both monoclonal antibodies and patients with convalescent plasma to respond to the mutant viruses. So this is this 501YV2 South African lineage. So these 3 monoclonal antibodies, they could perfectly well bind to the RBD of the wild type virus, but not this one. And when you look at neutralizing assays, these monoclonal antibodies could effectively neutralize virus, but they cannot neutralize these particular variants. So this is really concerning. And this is not the only paper like this. There's another paper from China, and it shows a similar kind of thing. So again, they looked at both convalescent plasma and a panel of monoclonal antibodies. And this is just some data extracted. So again, this is in vitro data. It hasn't been peer reviewed, but what you can see clearly is there is a difference between the wild type and the variants in their response to these monoclonal antibodies. So certainly these mutations are enabling immune escape from antibody neutralization. So this is bad. So there was a study quite recently in Nature Biotechnology, and it was a mouse model, and they basically did some very nice structural biology, and they developed non-competing monoclonal antibodies to bind to different targets, different epitopes on the receptor binding domain of the spike protein. And they showed how one antibody binding had an effect on the next antibody binding. They hypothesized that antibody cocktails might be more able to prevent this mutational escape, which we are already seeing with COVID. But the downside, that's a hypothesis. And the risk is that obviously the previous 2 studies I've just mentioned, they didn't just show data with monoclonal antibodies, they also showed that there was immune escape from convalescent plasma. So maybe we could develop highly specific monoclonal antibodies, a cocktail of however many, 10, 20, who knows how many we need. So then this comes back to the fact that the immune system is really great in that we make a diverse immune response and we have lots of different antibodies that bind to one virus. So maybe we can try to mimic that. What else is bad? Well, there's something about the infection. So if we actually think, if you notice that most of the antibodies that I showed you were IgG. So IgG is, you find a lot of it in the serum. It's easy to measure. It travels into the spaces between cells, but it doesn't go necessarily into the airways. So what do we know about the biology of how SARS-CoV-2 is spread? It tends to infect the apical part of the epithelial cells and shed and move down the airway. It's not getting into the blood, into the basolateral. Okay, so when we diagnose SARS, we don't look in the blood. We don't see virus in the blood, do we? What we see is we see virus in the mucus, in the secretions. We sample the nose, the back of the throat. You know, we want to get the saliva. So, you know, we really need to be thinking about where is the you know, the virus is coming in at our barriers, the alveolar and the bronchial epithelial cells. So if we look at the antibody that's there, it's IgA. So IgA, we don't really find very much in the blood, in the plasma, but we find a lot. In fact, you have more IgA in your body than you have IgG, but in order to measure it, it's just a bit more difficult. So this is what it looks like. So it's made by the plasma cells in the basolateral, in your mucosal organs. And then the antibody, the dimeric IgA, binds to a poly-Ig receptor, and it goes through this trans endocytosis. And the receptor becomes part of this, what we call the secretory chain. And this enables the IgA to survive in the harsh mucosal environment, because a lot of pathogens will make, as virulence factors, they will have enzymes that break down this IgA. So obviously, destroying IgA gives them a survival advantage. So this is the same thing again, just showing you that in real life, your IgA in your lungs and your gut looks like this. So maybe we need to produce monoclonal antibodies that are IgA, that are somehow protected from degradation, and that we deliver them to the mucosal surface, as opposed to giving them through the systemic route. What about the ugly? So antibodies are very expensive. There's a lot of research and development costs. So I was really interested to come across this report from the Wellcome Trust quite recently. And basically, 80% of the antibodies are used in a very small part of the world. 20% is the rest of the world. So Wellcome have come up with this global call for action to try and spread the benefits. So antibodies are very useful resources. They are the magic bullet of science fiction, but they're very expensive, and they're not equally distributed. And then just the last thought, in order to get better from any virus, and particularly from this virus, you really need to make your own immune response. And as part of that immune response, neutralizing antibodies is just one part. You will need your innate immune response, your interferon response. You'll activate natural killer cells. The interferons will activate your helper T cells that will ultimately activate and differentiate your cytotoxic T lymphocytes. So you need those to kill the virally infected cells. So neutralizing antibodies will block entry of virus into new cells. So there's two ways that you can generate this. You can generate this either by natural infection. Now, as we know, COVID has caused over 23 million known cases and over 2 million deaths. I have stopped looking at the data, it's so horrendous. If you told me last year that we'd already be vaccinating, I wouldn't believe you. So I think, hopefully, at the end of the day, antibodies will just hold things at bay if we passively transfer them. They have a half-life, they'll only last a certain length of time. But ultimately, let's hope the vaccine will be successful. But these treatments may be valuable early. They may be valuable for those more vulnerable, so the 20% that get severe symptoms, who are older, who have other morbidities, and particularly may be useful for patients with immunodeficiencies. So there definitely is a place for polyclonal antibodies, as in convalescent plasma, and hopefully for the future for monoclonal antibodies. But we have to be aware that viruses mutate, and the benefit of our immune system being so diverse is that if the virus mutates, because we make a polyclonal immune response and we're all different, then hopefully we can get around that. That's our way of fighting it. So good luck, everybody. Carry on fighting the fight, and thank you for watching this presentation.